Greetings, my fellow Earthlings, and welcome to Ask an Astrobiologist, the show for those who are curious about astrobiologists and what we do in our science. My name is Graham Lau, and some of you who are returning in our audience might notice that you haven't seen my face before. That's because this is my first time hosting the show. From here on out, I'll be sharing in the hosting duties with Sanjoy Sam. Our show is sponsored by the NASA Astrobiology program and the nonprofit Blue Marble Space. You can ask us questions on the show using the hashtag AskAstrobio on Twitter, by joining us in the chat room on seganet.org, or in the comments section on our NASA Astrobiology Facebook Live page, where you can also watch the show. Before we get to today's main show, we have to talk a little bit about our photo contest. You might notice behind me this really awesome picture of a cool location somewhere here on the planet that is astrobiologically relevant. Well, every month we show one of these. Last month's picture was a really cool looking image from somewhere special on the Earth. Now, yesterday we opened up the contest to see who could tell us what this location actually was. Well, we had several very good answers. We picked 3 of the best ones for who answered correctly. This is the Danakil Depression, the Dalaw Hot Springs in northern Ethiopia. These hot springs are rich in chlorine and sulfur. They deposit a lot of minerals around this geothermally heated fluid. And now astrobiologists are studying the organisms that thrive in these spring fluids to try to better understand how life can exist in extreme environments on Earth, how life might have originated on Earth, and whether or not we can find life in other worlds. So of all of our correct answers, we chose 3 as our top winners. They are Mahmoud El Said, who won first place, Grinch Hands on Twitter, who won second place, and Johnny O'Farnan, who won third place. Our third place winner will get some NASA stickers. Our second place winner gets some NASA stickers and some graphic histories of astrobiology. And then our first place winner, Mahmoud, you get some stickers from NASA, some graphic histories of astrobiology, and a very cool SegaNet drinking glass with the logo for SegaNet itself. So congratulations to all 3 of you. And for anyone watching now, tune in next month. Within a day before our episode, we'll announce the location competition to tell us what this site is right behind me right now. Now, before we get to speaking with today's guest, I should mention that the Danakil Depression in northern Ethiopia exists in a really interesting place on our planet. It exists in what's called the Afar Triangle, a region where the East African Rift Zone is meeting with 2 other mid-ocean spreading centers from the Gulf of Aden and the Red Sea. And those 3 are coming together and kind of spreading the continent apart, and the land is dropping, and that's why it's called a depression. This is also one of only 2 places on the entire planet where mid-ocean ridges spread right at the surface and can be expressed for us as geologists, which is what I am, to look at the surface of our planet. The other place that happens is in Iceland, the land of ice and fire. And today's guest happens to have been there several times for some cool astrobiological research that I think we'll probably end up talking about a bit here today. So without further ado, let me welcome today's guest, Dr. Amanda Stockton, an assistant professor in chemistry and biochemistry at Georgia Tech. Dr. Stockton, hi, and thanks for joining us on the show.
Hi, thanks, and thank you for inviting me to be here. I'm really excited to participate in this with you.
Oh, that's really great to have you here. I'm really excited for it. Again, my first time hosting, so it's kind of cool. I've always nerded out on the side and watched these episodes and thought how cool it is to hear about all the awesome research going on. Those who do know me know that I'm also involved in SegaNet, and I often answer questions for those who are high school students and college students who are interested in becoming astrobiologists themselves. And so I thought before maybe getting into your current research, it'd be really awesome if you could just tell our listeners about your process, your journey into becoming the scientist you are today.
Thanks. That's a good question. I was always interested in space, and I always wanted to go into space and look for signs of life. I grew up on Star Trek and Star Wars, and just the whole idea of being able to find life beyond Earth was incredibly exciting to me. Now, of course, I grew up on a cattle ranch in Oklahoma, and my dad had gotten an associate's degree, but neither of my parents really had much in the way towards education in the sciences, and so we really didn't have a good idea how to go from, elementary school kid that likes to play space cowboys and aliens versus a future where I could actually do that. So with time, that evolved. In 7th grade, I had a English teacher who gave us this fun assignment where we had to plan out our life and for the next 10 years. And so I really took that as a challenge. It was a report that we were supposed to write, but it was done in such a fun way. And so I looked in the National Geographic and Scientific American, and I found this article about Jennifer Harris. She has since moved on. Her name is now Jennifer Chosper, but she still works at JPL. And how she got there in this little bio is went to MIT, majored in aerospace engineering, works at JPL, sends stuff to Mars. She was the lead systems integration engineer for the Pathfinder mission, which was just way too cool. So that's what I wrote in my report. I was going to go to MIT, major in aerospace engineering, work at JPL, send stuff to Mars.
Very cool.
And for the most part, that's how it happened. I had a really great chemistry teacher in high school, and so when I got to MIT, I wound up double majoring in chemistry and engineering. And honestly, that was probably the best thing I could have done for what I actually wanted to do. Without background in both, I wouldn't be as well suited to the types of work that I get to do now.
That's awesome. It sounds very hard too, chemistry and aerospace. It sounds like a lot of classwork.
I'm sure it was. I remember more of the fun things than the work.
Oh, that's wonderful. That's very cool. It's cool that you were inspired at such a young age too, to kind of follow that kind of pathway of pursuing aerospace and the sciences, engineering and science together in your own career.
Well, at the early age, I didn't know what I was doing. I wanted to be an engineer. But later, when I got to MIT, it was just the chemistry classes were fun, the aerospace engineering classes were fun, and many undergrads do that like major of the month rotation their freshman year, and I kind of did a little bit of that also.
Okay.
And just wound up with enough credits to get both.
Awesome. Well, that's very cool. So then what was your pathway then into the research that you've done? I understand you have a master's degree and a PhD, and then you did some time as a postdoc at JPL, correct?
Right. So I did a master's at Brown. I graduated relatively soon after 9/11, and so there weren't very many aerospace jobs relative to the number of aerospace engineering graduates that were coming out. And so the market was really saturated over there. So I applied to a whole bunch of different positions, jobs from the aerospace side. And I also applied to a whole bunch of different chemistry grad schools because either or would have been perfectly fine with me. The program I got into at Brown, it was more actual chemistry, synthetic chemistry with a little bit of an analytical chemistry. I was developing a method using these small peptide chains that were bound to determine chirality of different species. The whole idea was that it would be a faster, cheaper way to determine enantiomeric purity of drugs, because it turns out that with many drugs, they are chiral. That means they're non-superimposable mirror images, just like your hands. And so just like when you go to shake hands with someone, if they offer their right hand and you offer your right hand, it lines up and you shake well. But if you offer your right hand and they offer their left hand, it doesn't quite work out right. And when it comes to handedness all the way down on the biomolecule side, if you shake hands, you have an effective treatment. If you don't shake hands, you have a poison. So that was kind of the thinking there. It turns out that I was not horribly enthused about pharmaceutical chemistry research. I find it to be incredibly valuable, and I'm really glad many people want to do it, but it was not my passion, and it was really challenging for me to really want to do it. And it turns out with many people, if you don't really want to do it, then you're not going to do as good of a job on it as maybe you could. So after a couple of years of that and doing some real soul searching, I applied again to other graduate programs where I could do more astrobiology type work, staying inside of chemistry, but pulling back in some of my engineering background. And that was how I found Professor Rich Matthews at the University of California, Berkeley. And that's where I did my PhD, working on the Mars Organic Analyzer system, which at the time was supposed to go on the ExoMars rover. It got de-scoped in 2009, which led to all of the funding being cut, which led to me graduating in 2010, but getting a NASA postdoctoral program fellowship so that I could continue some of that work down at JPL.
Okay, awesome. I mean, it's unfortunate that got scrapped from the ExoMars mission, but I'm sure it was still valuable for you to learn that process of what it takes to develop an instrument and then use it on a rover on another planet. I'm sure that process takes a lot of work.
It's absolutely fabulous. The instrument development process is very fascinating, and it's one of the things that I absolutely love.
Awesome, that's wonderful. Yeah, and then JPL, for those who don't know, is the Jet Propulsion Laboratory out in California. It's become kind of a mecca for space exploration. A lot of us space nerds love visiting, and it's really cool that you had a chance to work there. I'm so jealous. What was that like?
It was fabulous. It's one thing to get to do work that you're interested in. It's another thing to get to do work that you're interested in, and then on your way to lunch, just happen to walk past the engineering mock-up for the Curiosity rover that they're taking up to the Mars yard to test the maneuvers. Just all of the history and really cool facilities, and the work that's actually happening there is very exciting.
That's awesome. That's very
cool. The people are pretty great too.
Yeah, so I've heard. A lot of my friends work at JPL, and I'm quite jealous of all of them. It's very cool. So I understand that a lot of your time then, since your PhD, has been focused on developing instrumentation for surface experiments, but also for understanding how we can kind of mix those surface experiments with remote sensing as well, to better understand what we're looking for on these planetary bodies. Can you speak to that for a little bit about this process of developing instruments for various worlds and trying to look for life or signs of life on these worlds?
Yeah, so there's something called technology readiness levels, and that's TRL, that go from 1, you can write it on a napkin, all the way up to 10. We've flown it multiple times already. My group is really interested in that 1 to 3, where the exciting new developments happen that gives us access to a lot more different types of technology. At JPL, they're very good at getting you from 6 to flight. So once you have already done your crazy idea stuff, going from here the rest of the way up, but there's a little bit of a gap there. And that's where Berkeley Space Science Lab comes in. And so they're working on the Enceladus organic analyzer that was based off of the Mars organic analyzer that I worked on when I was a PhD student, and getting it from 3 to 6 over the next few years. And I've got a grad student that's helping with that project. Down here in the 1 to 3 range is where I'm taking that basic sort of technology and modifying it and making it more robust such that it could survive a 50,000 G impact with an icy moon. The acceleration that you get when you go to a dead stop from 5 kilometers per second within about 20 milliseconds of hitting an icy moon. And so it turns out that that's the accelerations that you would experience if you didn't slow down at all after a standard Holman transfer, which is the most efficient way to get from the Earth to an icy moon. You do a burn around the Earth, do another burn to go into an elliptical orbit around the sun, and then do another burn to slow down and go into orbit around your like Saturn or Jupiter. 5 kilometers per second. Slowing down from that is really hard. The Europa lander is going to be a very big mission. It's going to get to land in one place. But for the cost of one big mission, we could fly several little guys that can all hit the surface in a bunch of different places and get us a lot of different measurements. Obviously, this is not ready to fly on the Europa lander type of a platform yet. that's why we're down here in the TRL 1 to 3 regime, is how can we make these seemingly crazy mission concepts actually happen.
Definitely. Wow, that's really cool. I remember when I first heard about Europa Clipper, they had discussed the idea of putting some smaller like nanosats on the spacecraft and kind of launching those off the spacecraft to Europa. I know that idea has kind of been pushed to the side now, but it is a very cool idea for the future. I love that so much.
One of the problems we've had with doing these as landers is that we haven't had instruments robust enough to survive the impact. And so that's what's in here, is one of those.
Very cool.
But we're also interested in trying to figure out, if we did have one of these big landers, what would be the minimum instrument suite, the correct combination of measurements to accurately predict habitability of a sample, to figure out how many samples you must collect in order to actually represent a region, and kind of things like that. And for that, we've been in Iceland, where we do our Feldspar project. That's probably my largest funded project. It's a P-STAR, Planetary Science and Technology for Analog Research. And Feldspar is field exploration and life detection sampling for planetary analog research. Fortunately, the acronym actually makes sense, and it is the main mineral in our basalt samples that we collect in Iceland.
Very cool.
Yeah, and so we go to a bunch of very barren regions in Iceland. We've got 2 volcanoes, one Fimbulhals area. It was associated with the Eyjafjallajökull eruption that famously shut down European air traffic back in like 2010, 2011. We've been going to that site for ever since it cooled off enough to let people hike up there. So I think we went in 2013, 2015, and 2017. Although the group that came before me for the summer school that started this whole thing, I think they went in 2011 or 2012. I can't remember exactly. So that's one of them. And the other one, Holocrone, just cooled off enough to allow us to traipse all over that volcano in 2016. And we've been going there every year since then. And that's a very remote area. You have to drive for a really long time, bring a second gas tank, sleep in these little cabins that they technically have running water because there's a stream that runs behind them.
Yeah, remote fieldwork is pretty cool. It's really nice when you can travel somewhere so far away from people and find these really exciting spots on our planet to kind of explore. Can you talk a little bit more about your team, about the process? I know you mentioned the summer school. Can you explain how this kind of grew out of a summer school?
Yeah, so there's a Nordic Astrobiology summer school that was held in Iceland every other year, I believe. And then there was a winter school in the off years that was held in Hawaii. And it was a collaboration between NASA Astrobiology and the European Space Agency. Wolf Geppert was the organizer for the winter school in Iceland. And he had invited David Cullen, who was the deputy PI for the life marker chip, which just happened to also be on the ExoMars rover and was also de-scoped during that programmatic change in what was going on with the ExoMars rover. So we can commiserate on a couple of things. So Dave was running the science lab, Wolf was running the school itself, and a bunch of people, Diana Gentry, Morgan Cable, Adam Stevens, Gayatri Mirakesan, Eddie Schweiderman, Elena Amador. I know that I'm forgetting some people here. We're all part of that school. And after the school ended, they had fallen in love with the volcano. They fell in love with Iceland and the idea that we could do actual quality Mars analog research just as students and postdocs. So Morgan came back. She shared an office with me and a lab and said, oh, we're going to do this. Do you want to come? And part of it may have been that I had still MPP funds and could rent the car for the group. It may not have been totally altruistic, but we did. We put together bootstrapped funds from a bunch of different sources, and we went back out into the field in that 2013 field season. By the 2015 field season, I had just started my professor at Georgia Tech, and part of the startup funds I negotiated for was enough money to support one more field campaign to go to Iceland with all of my closest friends and collaborators. That turned out to be very fruitful, and so when we submitted to NASA after that, it became this large multi-year international effort. We've got a lot more new people involved now, like Erica Rader, who's now a professor at the University of Idaho, and a whole number of different students that have started picking up the early career side of this.
That's awesome. Could you mind describing some of the research that you guys are doing there? What kinds of instruments you're using in the field? What kinds of things you're looking for while you're actually there?
Yeah, so it is a P-STAR project, which means we're trying to have fidelity to a Mars mission, particularly looking at Mars sample return in three key areas. And one of those is the science, which I've already mentioned a little bit of how we're trying to figure out where to pick a sample. Does it matter if I pick it from here instead of here? What if it's 100 meters that way or a kilometer over there? What matters in those respects? We're also very interested in just this primary succession type of an event where after a whole life has been wiped out by a catastrophic geological event like resurfacing by a lava flow, how does life recover? What does that look like? And how do the communities begin to depend upon other members of the community rather than those little microclimate variables like the mineral composition, the moisture content, etc. So part of it is the science. Another thing is the technology, and you had asked about the instruments that we bring out. And so with the technology, we do have analogs to things that could be on Mars missions. So we do XRD, which is similar to what ChemIn does on Mars. We've got a ASD reflectance spectrometer, which is similar to what like CRISM is able to image from orbit around Mars. And basically, these are portable, handheld, bug wars looking laser guns that you literally zap the rock with a laser to find out what it's made of. I love it.
That's so cool.
After that, we pick up the samples and we drag them back to our field lab at the University of Akureyri, where we take the samples apart and try to figure out how much ATP is in them. That's adenosine triphosphate. And that is the molecule that all terrestrial life uses in energy. There's some thoughts that this is kind of an energy currency of terrestrial life and therefore can serve as a proxy for metabolic activity of the organisms in the sample. So we have to do that immediately because as you take the sample out of its native environment, some things are now going to freak out and die. Other things are going to go, oh, wow, this is beautiful. And they're going to bloom. And so doing that analysis as close to collection as is possible is really key in actually getting a good measurement there. After that, we drag everything back to the home lab where we do a bunch of other things. We do quantitative PCR for different primers that are conserved for different classes of microorganisms. We do community sequencing, moisture content, grain size, more XRF over at the University of Idaho. Just a bunch of different things, trying to kind of build this data set so that we can see what are those variables that correlate to each other, because ATP may not be the conserved biomarker on Mars or Europa, but the mineralogy should be the same. And so if we can kind of constrain what mineralogies help us get better habitability, then we may have a better shot moving forward in selecting samples.
Oh, very cool. Yeah, I mean, with Mars, we first see Mars from orbit, right? We do remote sensing looking down, and then we can send our landers, our rovers, to pick up samples and hopefully one day do a sample return and send them back home. And then I imagine what we would do here in a lab, either in orbit of Earth or in a safe lab somewhere here on the Earth, is to then analyze those samples in the same way that you're kind of doing right now, right?
Yeah, that would be the goal. And we were really trying to have this fidelity to a Mars mission on multiple scales, like what you're talking about. We wanted to have those analogs for orbit. We've got a quadcopter so that we can do digital mapping of an area. And there's been some talk of maybe putting a helicopter on the next Mars lander. We'll see. I think it would be a hexacopter, actually. And then down to those remote sensing type of instruments that could go on a Mars rover. So I didn't mention Raman. That has been selected for the Mars 2020 sample return mission. But we're also interested in those other instruments that are on the Curiosity rover and kind of having analogs for those. And then, of course, back to the home lab and all of the detailed chemical analyses that we can do here.
Definitely. That's so cool. I'm the director for logistics for a robotics competition that happens every year in the desert called the University Rover Challenge. And in that competition, we've actually had some students now build quadcopters, hexacopters to take off of the rover and fly out and do reconnaissance for them. And I can definitely see that becoming part of our future Mars missions as well. If that hexacopter or quadcopter from JPL does take off of Mars 2020, it'd be very cool. But let's step back for 1 minute. I do want to let our audience know, if you'd like to ask some questions of Dr. Stockton, you can use the hashtag AskAstrobio on Twitter to ask a question. You can use the comments section in the Facebook Live page, or you can ask your questions directly on seganet.org, where we're also live streaming right now. And so stepping back a little bit, we have actually one question already from our other host of the show, Sanjoy Sam. Sanjoy wants to know why Iceland is such a good analog for doing this planetary style of research, this astrobiological research.
Okay, that's a really good question. So Mars is quite barren. You don't see much green things growing there. And so in order for us to mimic that in any way, we need to go to some place that is also quite barren. Another thing is Mars doesn't have a lot of people traipsing around, and humans are kind of living, breathing bags of bacteria and just spreading our nasties all over a place. And so if you will see any pictures of us in the field, we're usually face masks, gloves, arm covers, and things like that to try to keep our human biota from contaminating our samples. And so one of the problems that we have with some of the different volcanoes around Hawaii and stuff like that is, even though it's new lava, it's also often right in the middle of this tropical paradise with lots of tourists and trees and birds and everything else. And there aren't too many trees or birds or humans to be shedding their bacteria over the volcanoes that we study in Iceland, particularly not Holocrone. Vindvalahals is a well-trafficked mountain pass now, and it is it's getting to be a little inhabited. And not only that, but we saw like multicellular life last time, lichen. So our volcano has grown up, and now its microbiota is determined more by the structures of the macrofauna that's providing nutrients to the soil.
Definitely. Yeah, it seems like everywhere we go on the planet, there's life. Earth is surrounded by it, right? Even in the most inhospitable environments, we still find living things on our planet. So it's definitely hard to find places without it, right?
Yes, there's even microbes that live in the nuclear waste, the nuclear water cooling systems.
Absolutely, yeah. One of my favorites, Deinococcus radiodurans.
Yep.
Its name means terrible little berry. Let's move to some questions now from our audience. Starting in is a question from Elizabeth Hutton on Twitter. She says, "Hi, Dr. Stockton. Your area of astrobiology is pretty much what I want to be doing with my biochemistry major. I've also been considering minoring in geology or oceanography. What are your recommendations?"
Okay, I did not take geology until I'd finished my postdoc and I took the class at a community college. I had to pick up an awful lot of geology along the way, and I would highly recommend just one class if you can in geology, because it's another language and it's not exactly the easiest thing to pick up. You're already doing chemistry. Chemistry is also one of those. It's another language things. And this thing about astrobiology is it is so interdisciplinary and multidisciplinary that just being able to speak the language can really help you out. You don't have to minor in it necessarily, unless that's something you're really interested in. But just getting the background on there is quite useful. I don't know what type of astrobiology you really want to be doing. You say it's the type that I'm doing sounds like the cool part. In which case, I would say getting some engineering background can be really helpful, because engineering is another one of those. That's a different language. And being able to speak science to scientists, engineering to scientists, and science to engineers is a really good skill to have, particularly if you want to do instrument development. I hope that helps her out.
I imagine so. I'm glad you mentioned, like these different languages we use. I mean, we're still speaking the same language, but we have different jargon and different ways of expressing our ideas in these different sciences, from biology and chemistry, oceanography, geology, aerospace engineering, and across the board. So I'm very glad you mentioned that. That's really important, I think, for our audience to hear. Our next question comes from Tom Caruso. Tom says, Dr. Stockton, thanks for your time. Can you take a minute to explain the possible seawater chemistry for a subsurface ocean, like what may be under the ice of Enceladus? Possible description of how a hydrothermal plume might behave in the low gravity of a moon like Enceladus.
All right. So that is getting a little bit into more planetary physics. Remember, I only took one geology class after I finished my postdoc. I can do the best that I can here. And I would strongly refer you to, there was an Enceladus special issue of Astrobiology recently, where the chemistry that was determined by Cassini flying through that plume was laid out in quite some detail. Here's what we kind of know at this point. We're relatively certain that subsurface liquid water ocean is in direct contact with a rocky core. Everywhere that we know of, i.e. Earth, where we have water interacting with rock on a planetary scale, you get subduction of the water down under the crust. That leads to oxidizing reactions of the rock, which reduces the chemistry in the fluid. Those reactions are exothermic, which means they release a lot of heat. And so that heat then heats up your fluid and forces it back out, and you get what are now known as alkaline hydrothermal vent systems from these sorts of environments. Those are high temperature, but they're actually pretty low temperature compared to like a black smoker volcanic system. So black smokers are magmatically heated, and they get up to like 450 degrees, and they can have lots of acid, and et cetera, et cetera. A alkaline vent is only in the 100-ish range Celsius, which we know of organisms that can survive above 100 degrees Celsius, thanks to the black smokers. And these are off-axis systems, so they're not magmatically heated, so you don't have to have active magmatic processes, completely driven by serpentinization type reactions. Everything that we've seen in the plume of Enceladus is consistent with there being active, ongoing alkaline hydrothermal vent activity. That would be really cool. There have also been organic molecules detected in the plume, and with some suggestions that perhaps we're seeing fragmentation patterns of larger organic molecules than the Cassini ion neutral mass spectrometer was ever designed to accept, because it was supposed to fly around in space and detect small things. It was never even, really hoped that there would be this massive plume shooting free samples out into space from this interior ocean. Now, how those systems might work under reduced gravity, like you asked, that would be a question for a planetary physicist, because I don't have a good handle on that myself.
Yeah, awesome. I recall some years back I attended a meeting at NASA Ames. We were considering whether or not Europa has plumes. As you might have heard, there have been Hubble Space Telescope observations that suggest there could be intermittent pluming of water out of Europa as well. And at that meeting, we were really talking about what instrument could we put on the Europa Clipper, for instance, that just in case there is a plume and we can fly through it, it'll help us out in figuring out what's going on. Because like you mentioned with Cassini, we didn't know until we got there to expect the plumes in the first place. So I'm wondering if we ever have another mission go back to Enceladus, and you were given the chance to develop an instrument for flying through the plumes and doing some kind of detection, what kind of instrument would you propose to send along on that mission?
Okay. Thank you for the opportunity to shamelessly pitch an instrument technology that I think has the highest chance of success. Sorry, my language just totally went out the window. You got me excited about instruments. There's something called the Enceladus Organic Analyzer. It's being developed out of the University of California, Berkeley, and the Berkeley Space Science Labs. It's based on microchip capillary electrophoresis. I'm going to see if I can show you this here. Do you see these long channels here? Those long channels are microfabricated capillaries, fabricated via standard microelectronic type processes, the same that we would do to make cell phones. Electrophoresis just means separating molecules by applying a high voltage. And so if we apply 15,000 volts across the length of that channel, we can start to separate molecules based on their charge to size ratio. And if we add in a chiral additive, we can start to separate the two hands. And from what we can tell, biology almost always has to do that one-handedness, just like I was talking about with the pharmaceuticals, where one hand shakes hands correctly, you get a drug, the other hand doesn't, you get a poison. That seems to be ubiquitous across biology. It seems to be something that is an evolved characteristic, no matter which hand is selected for at the beginning. Okay, so this separates molecules. Then we have a laser-induced fluorescence detection system. Basically, we zap the channel with a laser, and we look for the fluorescence of the organic molecules. To help in this, we have a compound-specific fluorescent molecules so that we can tag different molecules with these fluorescent probes. So we're not relying on native fluorescence. Instead, we've added in our own. And laser-induced fluorescence detection is quite simply the most sensitive technique you could possibly send. It's concentration sensitive. So an emission profile that would support the Enceladus Organic Analyzer, we would have a capture plate, we would fly through the plume, we'd close it up, we'd dissolve molecules, put them over into our microfabricated system where they're separated, the different enantiomers are resolved from each other, and then detected all the way down to sub-parts per trillion, lower limits of detection with the fluorescence system. That's the one I think has the best chance. We should be TRL 6 by the end of the next 3 years of the grant. And, if anyone out there is listening, please put us on your mission. That would be awesome.
Wow. I hope so. That sounds incredible. I really hope if we have an Enceladus mission in the near future, that we'll see that organic analyzer on that mission. That'd be so cool. It'd be really cool to see some of the images of the data coming back as well, showing us if we're actually making those fluorescent detections. I would love to see that.
Before we go on, PI on that one is Richard Matthews. He's out of the Space Science Lab. I have to make sure that I'm clear about that and not inadvertently taking credit for all that work.
Awesome. All right. Our next question comes from SeganNet from Siddharth Pandey. Siddharth asks, what are some of the pressing engineering challenges faced during feldspar that would potentially be a concern during off-Earth exploration?
Oh, that's a good question. Most of the technology challenges that we experience are either, I would categorize them into being kind of like a logistics problem, and the other being kind of like a stupid problem. The stupid problem is you have everything with you that you need, but you forgot to pack the batteries. That is on an actual space mission, you would have a list of requirements, and then you would have your validation and verification engineer come in and check that all of these requirements have been met. And so that's a big team effort where everyone's going through and checking, like, yes, we brought the batteries. Everything is charged. Everything's going to work. So the stupid problems, we have mechanisms in place to avoid those in an actual mission, and we should have better ones for our own work. We do have checklists, and we are getting better about this. There's always just that point, like late in the campaign, when everyone's like, oh, we've done this a million times, we don't have to go down the checklist. Yes, you still have to go down the checklist. And then over here are more logistical problems where, for example, getting a target of opportunity and wanting to go collect a bunch of samples from that spot, but since we did not plan ahead for it, all of a sudden we don't have enough consumables in place to do the sample collection and analysis without kind of calling in support. And you can't call in support on a mission. But the problem is even more intense on a mission because consumables are, for me, it's an extra checked bag. You can't just check an extra bag with a Mars lander. So those types of concerns, those become a really big challenge for a mission team to figure out whether that cool target of opportunity is actually worth going to pursue, because it might mean that it's limiting our future science over in this other area that we wanted to do. And those are big discussions held by lots of people on large teams. So it's not one person trying to figure it out.
Absolutely. Yeah. It's interesting how collaborative the science gets with those very large scale missions like that, then, where there is a lot of input from a lot of people.
Yeah.
So thank you for that answer. The next question is from Richard Gordon, asking on SaganNet. Richard asks, starting from a barren volcano, in what order of succession do you see bacteria, archaea, and single-cell eukaryotes?
Oh, that's a really good question. So, Richard, we're trying to understand that. So far, our tools and techniques are somewhat biased, so we see a lot more bacteria than we see archaea, and we see more eukaryotes than we see archaea, but bacteria dominate. That doesn't necessarily mean that bacteria do dominate. As we're learning, as we go through the lab process and optimize our protocols, we're finding out that our bacterial assays are just a lot more efficient than our archaeal assays. And part of that might be that bacteria has been studied more by geomicrobiologists than archaea historically, and only recently have archaea really came into this new hotness of wanting to know more about them. And our eukaryote assays are also just a little less efficient, it seems. We're not sure. We're still trying to nail that down. Actually, I think that Scott Sutton is out there working on that today, right now. So it's a good question. We want to know. Do we actually have a good handle on the answer? Not entirely, but we should really soon.
Oh, that's wonderful. And I look forward to hearing more about the results from Feldspar then for what we're going to learn from that. Our next question comes to us from Twitter, from user Avi at vid098. Avi asks, what suggestion would you give to a graduate student in geology who wishes to be an astrobiologist and pursue a PhD? What would be the right courses to take?
So you're a geology person in grad school already. Good courses. Mike, should I keep going or should I pause?
Let's keep going, Amanda. Thanks.
Okay. So in geology, you probably have not gotten very many biology type classes, but you probably had some chemistry classes. So you probably can speak chemistry okay, but biology is going to be a bit of a stretch for you unless you make the effort to jump out there and get those classes. Other ones that are available to you probably inside of your own program would be the planetary science type classes. And obviously taking those would be fun and useful. But given what you said about your background, I would suggest biology, biochemistry as some areas that you're probably not getting the kind of that language background inside of your current major.
Awesome. Yeah, thank you for that, and I hope that helps Avi. Our next question comes to us from Stephanie Colon Santos. Stephanie asks, could the mineral evolution theory, as proposed by Bob Hazen, be a starting point for a generic biomarker, so not necessarily ATP dependent, Or is the ambiguity of the planet's evolution a major bottleneck for detecting life as we know it?
Okay, so first off, I want to address the ATP as a biomarker. I am not suggesting in any way, shape, or form that we go to Enceladus and we look for ATP. I'm simply saying that given that feldspar is constrained to Earth and we are looking for Earth life, that this is a very easy biomarker for us to use here to ascertain habitability here on Earth. Once we start talking about going to another location, that's when these types of molecular evolution theories come in quite handy, where we can realize that the set of molecules that life uses has been evolutionarily selected for, given the unique chemical and thermodynamic parameters of the environment in which that life evolved. Now, on Earth, that gave rise to 4 nucleobases, a set of 20 amino acids that we use in our proteins, and it's all homochiral, one of those hands. We can then make some assumptions that if we were to go to another location, if life had evolved there, it would similarly have narrowed down the vast suite of organic molecules that you would find in like Titan's atmosphere or meteorites, where there's just hundreds of amino acids, racemic for the most part, lots of different nucleobases. And so that like broad mix of different organic molecules, that doesn't look like life either. What we want to do is we want to go and we want to see where the number of different molecules has been limited to just a few. And that's kind of the paradigm that I'm looking at. And I think that that's falling in line quite nicely with what you're recommending as well, which is not stick to a single biomarker. We're not going to look for ATP. We're not going to look for cytochrome C oxidase. Those are the evolutionary end products. And what if that life selected a different track for evolution and never got to ATP, got to a completely different energy currency molecule? Yeah, so that's my soapbox on that one. I really can't get behind looking for a single biomarker or a single class of biomarkers. I really think we have to have a broad search where we can survey the entire chemical environment.
Absolutely, that's very cool. I think a lot of us in the astrobiology realm have kind of started opening out that way too. Ever since, the 70s, when we sent the Viking landers to Mars, we were looking for a very specific kind of Earth life. And there's a lot of other potential out there. So I like your suggestion a lot of kind of keeping our minds more open to the possibilities, because life might not have evolved the way we did. I love that a lot. Our next question comes to us from Sanjoy Sam. He wants to know if you can speak a little bit about the mentoring process. If there's anything that you know now that you're a professor and you're mentoring students of your own, if you can speak to the process of being a mentor and also of finding good mentors.
Okay. I don't necessarily know that I know how to be a good mentor yet. I'm still working on that. I'm sure my students can point to a number of mistakes that I've made. In terms of finding a good mentor, I saw this TED Talk the other day where the message was basically to teach bravery, and that society has traditionally rewarded bravery in boys and performance in girls. And so that leads to, at these higher levels of education, now we've got the group selected that have been very high performers. They've been rewarded for being high performing, but they haven't always been rewarded for taking risks, for going out on a limb. And so I think when you are going to find a mentor, it can be very scary, and it can seem like you're really extending yourself and you're putting yourself out there. And those people who have been conditioned to just perform well and not extend themselves out are going to have a lot of trouble doing that. But the best thing you can do is walk up to that person, call that person, say, hey, you have the job that I want to do. Can we talk? Like, I'm looking for a mentor. You will never find a person who is genuinely interested in their job who is not going to be like, oh, yes, let's talk. let me tell you all about me and my cool work. So if you don't get that sort of a response, maybe the person was busy. Maybe you should try again. Or maybe the person is not the sort of person you want as your mentor anyway, and you should go try the next person. So that being brave and being willing to jump out there and take a risk and build your mentorship team to meet your needs rather than help provide that positive reinforcement of being a high performer. I think that's a very important message that many students need to get. And I've seen several students kind of limited by their desire to always come across as the perfect student rather than being willing to be like, hey, I don't know what's going on. Can you please help?
Yeah, I definitely would have made it through grad school if I didn't have those moments where I could just ask a question and be like, I have no idea how to do this. Can you help me? I also love that idea of finding your bravery. I'm a Toastmaster, and I often tell people when learning how to do public speaking is to find comfort in being uncomfortable. And so it kind of goes along those same lines of finding your bravery. I love that message a lot. Let's see, we have some time for a few more questions yet. On SeganNet, Penny Boston asks, if you had your choice and money being no object, would you rather have a single rover with major instrumentation on it, so like another big curiosity, or a suite of mini landers/rovers, but with each having much simpler instrument capabilities?
All right. So Penny, can I have both? And thank you. I suppose it goes both ways. It depends on the instruments that you can put on either of them. In my personal experience with fieldwork, you get exceptionally different environments in different locations, and it's hard to access all of those different environments with a single large rover. And so it would be so valuable to be able to get access to those multiple other environments. However, without the right technology on those little rovers, the information you're going to get back is possibly not worth sacrificing your big rover. So I want both, and that's why we're doing the little easy to fly, easy access to surfaces type instrumentation, because we really need to boost the capabilities of these non-flagship class type instrument technologies so that we can have several little rovers that are basically the equivalent of one big one. But
absolutely, yeah. I would love to see the big ones and the small ones and do it all. Let's just do it all. Give us all the money and we'll just do everything, and that'd be awesome. I have a few more questions from the audience, but I apologize to Pritha, to Brian Collins. I apologize. I'm going to ask the last question myself, actually. I noticed on your website you have a quote from Elon Musk that says, "I believe it is possible for ordinary people to choose to be extraordinary." Can I ask you why you put that quote on your website and what that means to you?
So I think I just found that kind of randomly. I was looking for a good quote to put, but it kind of meets this whole idea that you get to build the life that you want to have. You get to go out as a geologist and find your biology classes, as a biologist and find your geologist classes, and you get to pick your mentors and pick the people that you will mentor. You get to develop your own community. And just this idea that a group of otherwise ordinary early career people could get together and start this feldspar project, this now multinational fieldwork initiative, that just kind of fits in line with the whole thing. I think it's important for people to remember you can feel so small and ordinary, but you can do extraordinary things when you want to. You just have to want it. You have to make it. You have to build it.
Absolutely. Get out there and do it, right? Dr. Stockton, thank you so much for your time today. We really appreciate having you on the show. You've been a wonderful guest. I apologize again to our audience, the questions we didn't get to today. We'll try to answer those sometime throughout the coming weeks on Twitter and Facebook. Again, Dr. Stockton, thank you. To everyone out there, ciao, and stay curious. Bye, y'all.
Thank you.